Okay, so we're in 7.6. We're talking about human chromosomes today. We'll start out on slide 60 with the Human Genome Project. Now, the Human Genome Project, it was set as a goal to generate the first sequence of the entire human genome. The reason behind all this was that we wanted to map where on each chromosome would be each set of genes. And so this provided fundamental information about the human body as a blueprint of where things are located, how things are then passed on from parent to offspring. And then we've actually been able to advance medicine since we've had this human genome mapped. The Human Genome Project was very significant because it was a world effort in order to get all this information mapped. That was a ambitious project starting in the 90s, and it, the goal was to map and understand all of the genes in the human species. It was completed in 2003, and essentially some key outcomes to come from that. The project as a whole successfully sequenced about 99% of the human genome. It helped to identify genes that are associated with diseases such as cancers, Alzheimer's, cystic fibrosis. It also accelerated advancements in personalized medicine. It allowed for gene therapy and things that we'll talk about later in this lecture, as well as innovations to forensic science and biotech as well. It also helped to develop the field of genomics and has improved DNA sequencing technologies in order to complete the DNA sequence for the entirety of the genome, which you're looking around 3 billion base pairs of the A's to T's and C's to G's. What are chromosomes? Again, we talked about chromosomes being these condensed strands of DNA and all of that information condensed into a chromosome. And that's important because we have different chromosomes within the human body, and we actually have two different types. We've got autosomes and we have sex chromosomes. So in the human body, we have 46 chromosomes total with 23 pairs. The pairs of chromosomes would be 23 in total, and those are sister chromatids attached at the centromere, and that would be 46 chromosomes total. 22 of those pairs, so pairs 1 through 22, those are called autosomes. Those are regular body chromosomes that it would be found in both male and female. And then we have one pair of sex chromosomes, and that would be XX in females and XY in males. And that's going to be important because we can tell based on something called a karyotype, which is a picture of the chromosomes. We can tell gender based off that image. So for human chromosomes, again, we have 46 total, or 23 pairs of chromosomes. The first 22 are called autosomes, and that's going to contain information both important to males and females. So they are named by the chromosome number, right? So if you're talking about chromosome number 1, that would correspond to a pair. So 2 number 1 chromosomes. You'd want 2 copies of the number 1 chromosome. Same thing would go from 1 to 22, and then number 23, that would be the sex chromosomes, which would determine scientific gender of the human. And so those are not labeled with numbers. Instead, they're labeled with letters, so they're given X or Y. So for a typical male, that would be XY, and for a typical female, that would be XX. When we look at sex chromosomes, the males versus females are going to have 2 copies of the X chromosome. On a karyotype that we'll see in a second, they are longer or taller, and so you can see if there are 2 tall X chromosomes that would be the female genotype XX, and you would write that as 46-XX. That would tell us that is a normal, typical female. As for males, we're going to have 1 copy of X and 1 copy of Y, and so that genotype would look like XY, and you'd write that as 46-XY. Now, the Y chromosome is the chromosome that contains the gene called SRY, which is going to trigger testicle development for an embryo. So females don't have the Y chromosome, and therefore they don't have the gene. for testicles to form. Instead, they'll form ovaries, and that's right away the difference there. Karyotyping is the picture of the chromosomes, and so this is going to help. It's used in lab to identify an individual's chromosomes, to detect chromosomal abnormalities, which could potentially be linked with a specific genetic disorder or disease. And so essentially how the collection works, you are looking at collecting cells from sources like blood or amniotic fluid or even bone marrow. They culture the cells that they have collected in a lab, and they chemically treat those cells to stop during mitosis at the process of metaphase, when those chromosomes are the most visible under a microscope. So they stain those chromosomes so that they can reveal the bands, the patterns that are on the chromosomes, which look like stripes. And then they arrange them in pairs by size and shape to make a picture, a karyotype. And so a karyotype can tell us a lot of different things, but essentially they can be organized into a diagram, if you will, where we can count how many chromosomes are there. We can look for chromosome abnormalities, like an extra chromosome or a missing chromosome or a structural change to the chromosome. And then that can tell us a lot of different things about the person. So this is used often to diagnose chromosomal abnormalities. The most common chromosomal abnormality in humans is Down syndrome, which is resulting when a human has 3 copies of the number 21 chromosome. So 3 copies of chromosome 21, which results in a total of 47 chromosomes. So when we look at this karyotype right here, looking at this genetically normal karyotype and trying to figure out, is this a male or female? I think that the first and most important thing that we know how to do are to tell which ones are the autosomes and which ones are the sex chromosomes. So for the first 22, if you were to label those and write out numbers, which I highly suggest doing. Okay, so if you labeled your chromosomes, you've got 22 autosomes, and you have 1 pair of sex chromosomes. And so if we're determining gender from this, what we can see is because that chromosome pair 23 is not exactly the same in height or in band pattern, we're going to call that XY. So this would be a 46-XY karyotype, which means that the phenotype would be a male. Okay. As for slide 68, same thing here, except that you're seeing for chromosome number 23 that those would be 2 X chromosomes, so XX. So if you were to tell me the genotype, that would be written as 46-XX, and that would mean that this person is a female. As for chromosome karyotype on slide 69, you can see here that we're missing the second chromosome for the chromosome 23, right? Okay, so for this genotype, we would write 45-X. And that 45 is important because we are denoting that we're missing a chromosome. And this actually, the diagnosis would be Turner syndrome. And if you want to know more about that, maybe you could pick that for your genetic disorder project that you'll see coming later. But essentially, Turner syndrome is this genetic condition that affects females, and it occurs when only one of those 2 X chromosomes is formed or is present. So this karyotype tells us quite a bit about that embryo. Looking at page 70, You might not notice right away, but go back and double check where is your autosomes. What are they doing? Okay, so a couple things that we need to get from this karyotype. First is going to be determining the gender. So if we were looking for the sex chromosomes, number 23, what's that doing? It's an XY. And then if you were going to numerically number your autosomes 1 through 22, you'd notice that we actually have on chromosome number 21 a third copy present. And so that third copy is going to be a trisomy, or third copy of 21. So this trisomy 21 would tell us that there are 3 copies of chromosome 21 instead of 2. Now, you might have heard this already when I said that there is a very common genetic condition called Down syndrome, and this is resulting from a third copy of chromosome number 21 instead of 2. And so to write this genotype, this would be 47-XY. Okay, and that's where we're going to end for today for the karyotyping lecture. Now, you do have a karyotyping IP to do with some stations around the room, where hopefully you'll learn more about different genetic disorders and how they present on a karyotype.